All right, let's talk about tertiary structure then. So that's the third level of structure. And by the way, these can be abbreviated. Primary can be like this. Secondary, just put like a little degree sign, a little circle. Tertiary, and then quaternary, which is right here. Tertiary is right here. All right, tertiary structure is simply the folding of the amino acid chain, including secondary structures, alpha helices, into a complex shape, into the final complex shape of the protein, which is seen here. So you see beta-pleated sheets involved here. You see alpha helices involved. Basically what you're going to get is some kind of a complex final globular structure of the protein that involves alpha helices, possibly twisting around on each other like this. You know, all kinds of different configurations. You might have some beta sheet regions. And it's hard for me to draw a three-dimensional aspect of it here, but you'll have both. Now, the other thing you might notice is that you might have regions of, where it's just primary structure, just straight chains like this also. Okay, I'm just randomly throwing stuff in here, so there's lots of different variations, lots of different. Oh, I'm missing a part of my protein here. Oops. Let's put that in. Anyway, so here we'd have some alpha helices. Here we'd have some beta sheets. We'd have some regions where it's just primary structure, just simply a chain, something like that. All right. The key thing about tertiary structure, though, is that it is held together. My goodness. It is held together with linkages between R groups. It's held together with linkages between R groups, which is different from secondary structure. Keep in mind, secondary structure was just hydrogen bonds between those atoms of the backbone. Okay, now these R groups that stick out, they're going to link together with a bunch of different types of linkages and then form that tertiary structure. All right, so what's going to happen here is you might have an R group here sticking out, you might have an R group here sticking out, and those might form some kind of a connection, maybe a covalent bond. You might have one sticking out here, you might have one here, and they're going to link together maybe with an ionic bond or a hydrogen bond. It just depends. And that's the kind of strengthening you're going to get then between these R groups in order to strengthen the tertiary structure, to strengthen that globular final shape of the protein. All right, so we should maybe talk about what these linkages might be. And in order to do that, I'm just going to cheat and go back and use this blank page I had from before. All right, so types of linkages in tertiary structure. So we're going to go from strongest to weakest. The first and strongest type of linkage is a covalent bond that we call a disulfide bridge. That's covalent, which is the strongest kind of bond there is. These happen between two cysteines. Because cysteine is one of the amino acids that had sulfur in it, and it can form this sulfur connection like this on the R group. Okay, so that's your disulfide bridge because there's two sulfurs involved, and that's the strongest. All right, but you need cysteine. Okay. Then the second strongest that you're going to see is an ionic bond, or ionic bonds, which are clearly going to happen between charged amino acids. So two charged plus and minus R groups might link together. And those are pretty strong too, but not as strong as covalent. Okay, but if you got enough of these, that forms a pretty solid, pretty strong connection. All right, so sometimes these are also called the acidic and basic amino acids, or R groups. All right, third strongest would be hydrogen bonds. Hydrogen bonds, okay? So hydrogen bonds are sometimes described as pretty weak, but if you get enough of them, they are significant. And these are going to happen between polar amino acids, between polar R groups. So any of those ones that we talked about before that had the oxygens and the nitrogens sticking out with those partial charges, they can form hydrogen bonds between each other. So these three are really the main way that tertiary structure is linked. But there are two more sort of less significant or much weaker kind of interactions. And four would be the hydrophobic interactions. And clearly those happen between non-polar R groups or non-polar amino acids. Any of the ones that have R groups that just have carbon and hydrogen in them on that big list, that first list that I showed you. And then finally the weakest, or actually these two are kind of equally pretty weak, are van der Waals forces. Van der Waal was a scientist, maybe Dutch or something, probably with the van in there. Van der Waals forces are just sort of fleeting, temporary partial charges that happen when the electrons orbit a molecule. And if you have a molecule here and the electrons are orbiting around these nuclei, sometimes the electron happens to be on this side and it makes a slight negative charge. So then if you have another molecule over here, it's going to cause a slight positive charge to form over here by pushing its electrons to the other side. All right, so if this electron, well, it's multiple electrons, but let's just talk about one. If this electron happens to be over here on the right side of this molecule, the left side of this molecule will then sort of automatically turn positive. And then as this electron orbits and goes to the other side, pretty soon it'll be over here, then this electron is going to follow also. And then this over here is going to be positive and this is going to be negative. All right, so those are really, really weak charges, or really weak forces, much weaker than hydrogen bonds. They're kind of similar in principle to hydrogen bonds, but hydrogen bonds are permanent. So these guys are permanent, they're always there. These guys are temporary and they kind of keep oscillating back and forth. But as they keep oscillating, they keep applying. It's not like the attraction goes away. The attraction stays because as this moves over here, the other one also moves and it continues to attract the whole time. It's just not very strong. All right, so that's the 5 from strongest to weakest: disulfide bridges, ionic bonds, hydrogen bonds, hydrophobic interactions, and van der Waals forces. I have not been reading the closed captions, but I'm curious how good the interpreter is when it comes to some of these science terms. Let me try one. Disulfide bridges. It said one. Disulfide bridges. No, I'd actually got it. How about cysteine? Cysteine. Okay, well, that's supposed to be cysteine. So, yeah. Van der Waals forces. That worked. Sort of. Didn't put the space in there, but that's all right. Okay, cool. I did not know PowerPoint had that feature. I just learned that, and I will try to include this in my PowerPoint so you can read along with me talking. All right. Okay, next, the next level of structure we're going to discuss is quaternary then, okay? And quaternary is real easy. Once you have a tertiary structure which forms kind of a globular protein, so here you got one, another one might link to that. So you have another one over here, another one over here. A lot of times it's 4, but not always. And these can then link together, and they link together with the same linkages that we use to link tertiary. So these 5 are really, the first 3 are most important. Will link quaternary structures together. So a lot of proteins have sort of subunits or smaller globular structures that then link together to form this sort of mega protein. One example would be like hemoglobin has 4 subunits, and those are linked together with those types of attractions and form quaternary structure. Some proteins are just one single molecule, where it's just one tertiary structure and that's it, and it's finished. But some have that higher level. All right, so this picture comes from Campbell from your textbook. I think the same one is in your textbook. and it shows you the different attractions you might see. So it shows you hydrogen bonds here between these partially charged ones. These R groups. It shows you hydrophobic interactions because these guys have just carbon and hydrogen. They're non-polar, nothing polar here at all. Disulfide bridges, and you know what this is, obviously. You know that's cysteine, right? Of course you do. There's another one here. Two cysteines can form that disulfide bridge. And finally, ionic bonds between acidic and basic or charged amino acids that we can see here. And this is a full-blown plus, a full-blown minus, so that's a pretty strong connection, that ionic bond. Quite a bit stronger than, let's say, the hydrogen bond, which is just a partial positive and then a partial negative. So we put the parentheses around that to show that it's just partial. It's a fraction of a charge. All right, so hemoglobin is an example. Like I told you, it's got the 2 alpha and 2 beta subunits. They're all kind of similar, but they have some slight differences. There's iron in there too, which helps carry the oxygen. That's the heme group. So we got 4 subunits making up 1 hemoglobin. Collagen is another example of a protein that forms quaternary structure where there's multiple tertiaries linked together. That seems to have a lot of alpha helices. All right, here's a better look at hemoglobin, a little bit more realistic, and it shows you the sequence for normal hemoglobin, at least a part of the gene up there. It shows you a part of the amino acid sequence. And then what might happen if there's a mutation, like what happens to tertiary and quaternary structure if you mess with one of those amino acids due to a single point mutation on the DNA, and that's something that's going to result in possibly something like sickle cell anemia. With sickle cell, we just simply took this one glutamic acid actually, and changed it to valine up there, the red one. This guy, where's my pen? My pen is gone. There it is. All right, and that causes a drastic change in the beta subunit in the tertiary structure, which then causes the quaternary structure to be completely wrong. It causes them to link together where they shouldn't. It has these hook shapes that now can link together and form these rods, and the rods cause a distortion of the red blood cells. They cause the red blood cells to form like a moon shape like this, or a sickle shape, which reduces their oxygen carrying capacity. All right, so normal red blood cell, here's how they're supposed to look, like a nice donut shape. And then sickle cell has that moon shape or sickle shape due to that one single amino acid being wrong, causing a drastic change in tertiary and quaternary structure of that protein, which is, interesting and obviously medically important to understand. Proteins can denature. So proteins will have some kind of a tertiary / quaternary structure if you do things to the protein that the protein doesn't like. Usually that means heat. It will become denatured. Other factors might be pH, salt, presence of salts, stuff like that. Generally they don't renature. This is kind of rare. Sometimes they can go back. Sometimes it's reversible and they can go back to the correct shape, but a lot of times this is permanent, where now it's broken and it can't come back. Like if you drop a coffee cup and it shatters, it's kind of hard to undo that and reverse the process. That's usually how it works with proteins. Like when you fry an egg, the protein gets the albumin and the yolk. Those proteins get denatured, and just by cooling it doesn't just bring it back. Some proteins need help in the way that they fold into their tertiary and quaternary structure, so they get help in the form of a helper molecule in the cytoplasm that's a chaperonin, kind of like a chaperone for kids on a field trip or something. But the chaperonin allows the protein to enter this sort of like a big container. A cap goes on, and then there's perfect conditions in there for the protein to fold just the right way. Then it opens up and the protein comes out ready to fold. And not all proteins need this, but some are so complex that they need a little bit of help to fold correctly. All right, that's it for proteins. Thanks for watching.